Today's topic is an introduction to microbiology. Let's now try to understand what microbiology is. So microbiology broadly is a branch of science that deals with the living organisms which are not visible to the naked eye. There were significant contributions made to the field of microbiology over time, like taking the example of Louis Pasteur, who led to the development of a vaccine for hydrophobia, that is the rabies vaccine. It is considered as a milestone in the development of immunization in medicine. Also in Paris, the Pasteur Institute was built by public contribution, and they mainly focus on the preparation of vaccines and investigation of infectious diseases. He is also regarded as the father of microbiology. Talking about Joseph Lister, he applied Pasteur's work and introduced various antiseptic techniques in surgery, and they significantly reduced the mortality and morbidity that was caused because of surgical sepsis. He is also regarded as the father of antiseptic surgery. Similarly, Robert Koch and Paul Ehrlich, they are regarded as father of bacteriology and father of chemotherapy respectively because of their significant contributions made in their respective fields. So as mentioned earlier, microorganisms are too minute to be visible to the naked eye. Not to visualize same, we need some instruments. And thus the concept of microscopy was introduced, which mainly has the following applications. It helps in the magnification of the object. It also aids in enhancing the resolution. It also helps in optimizing the contrast between the structures, the organisms we are studying and their background. And today various types of microscopes are available to us based upon their individual applications, including the light microscope, the fluorescent microscope, the phase contrast microscope, dark field microscope, electron microscope, and many others. Now, if one intends to study the structural details of a microorganism like a bacterium, it cannot be done using a light microscope because of the absence of contrast, and for same we need some staining methods. And some of the common staining techniques available to us are simple stains, impregnation methods, negative staining, or the differential stains. The examples of simple stains include basic dyes like methylene blue. In negative staining, we have the examples of India ink, and the Gram stain or the Albert stain or the acid fast stains all fall under the category of differential stains. And the microscopic examination of the bacteria reveals various types of shapes, including the cocci, the bacilli, the comma shapes, and so on. Overall, in microbiology, we study about bacteria, both eubacteria and archaebacteria, fungi like yeasts and molds, algae, protozoa, and viruses. Now, let's first have a look at the bacteria. The bacteria are prokaryotic in nature. There is no exception to this. And we have already studied the differences between the prokaryotic and the eukaryotic cells in our cell biology class. You can check the link in the description. Now, the bacteria have various types of shapes and sizes, but overall, they come in the range of 0.5 to 5 micrometers. And the shapes include rods, spheres, spirals, etc. If we talk about fungi, the fungi are eukaryotic organisms. They possess cell walls. They may be unicellular or multicellular in nature. The protozoa are single-celled eukaryotes. Examples include amoeba, and viruses like tobacco mosaic virus or coronavirus. They come at the borderline of living and non-living. That means they are active only once they come inside the host. If we talk about prions, they are smaller than viruses. These prions, they are infectious agents, and they are proteinaceous in nature and act as obligate parasites. They are responsible for certain diseases like the mad cow disease. That was all in this section. In the next part, we are going to talk about the pathogens and the pathogenicity.